Is my screen visible?
Yes.
Perfect. Hello, everyone. Thank you so much for this opportunity. I wish I could be there for the presentation, but I have started my next semester here. This picture here just gives a brief idea of the master program that I'm involved in currently. In short, it's called MEME. It's a mobility master in evolutionary biology, and all the different universities shown here together organize it. I did first semester at the University of Groningen, and for the next two, I was at the University of Montpellier. And during one of those, that is the third semester, is when I did the project with Karen and Rafael that I'll be talking about right now. And finally, I'm back to the University of Groningen, but doing an external project right now at the University of Amsterdam. So just to say it again, that my course is on evolutionary biology. Here's a picture of Darwin, of course. And the idea here is to show this text. It's a little too much, but what I wanted to say from this is that biodiversity is generally very varied across different regions of the Earth, but the climatal factors or the physical conditions of any of the different regions which are present are not as dissimilar, which means that any condition in one region can be, another region can also have such similar conditions, which means taxa or organism from one region can also survive in another if they can cross the barrier in between. And that crossing is facilitated by humans in these days. And this entire phenomenon gives a good starting point to talk about invasion. My project was on the invasion history of house mice in Senegal. One second. Yes. Biological invasion is basically the introduction, establishment, and subsequently spread of a taxon in a region to which it's not native. The graph that you see here shows the first record of an invasive species or of a non-native species in a region outside its normal range. And it's across different countries, but each year it's the first record rate per year. And you see a steady increase in this pattern with no signs of slowing down. And the increase has been the steepest in the last few years. One of the biggest impacts of invasive species is the native species extinction. A very well-known example in this case is the rosy wolf snail, which alone was responsible for extinction of over 100 species of native snails in the Hawaiian Islands. These quantitative figures, along with the fact that the most prudent option to prevent damage by invasion is to be able to limit future events, calls for better understanding of the processes that facilitate. Here, I talk about one such species or a subspecies of Mus musculus, the house mouse, Mus musculus domesticus. The species complex originated somewhere in Southwest Asia and has been a commensal on humans for about 12,000 years now. Currently, they are cosmopolitan, that is found everywhere except Antarctica. Their colonization history, as we can also see from this graph and in general from literature, it is better understood in Europe and certain islands around here, but there's limited information from Africa. The study tried to understand the colonization history of Mus musculus domesticus, house mice, in Senegal, which is one of the countries in that continent. This is basically what the map of Senegal looks like. And the first invasion of house mice happened in the port town of St. Louis. And this has been the only event of introduction of this taxon in the region. There was a subsequent secondary introduction in Dakar. And with increasing road network connections and commercial links, there has been an eastward expansion. Although this expansion started comparatively much later than the initial introductory events. The idea here was to see or compare two regions that we call the anciently invaded and the recently invaded. The anciently invaded region had individuals since about 100 years, while the recent ones, they have individuals only since last about 5 years or so. And in each of these regions, we have 3 localities or villages, because as I said, these are commensals and generally restricted within the limits of the villages. And because there was an eastward expansion, this is the invasion route is what is assumed. And we tried to compare what are the differences or similarities between the region that was anciently invaded and recently invaded. Specifically, we wanted to understand the demographic processes that were responsible for the spatial spread and compare the invasion history from a demographic perspective. The sampling was done in Senegal. I was not part of these. Sampling was done in Senegal within houses, and approximately 40 individuals were sampled in each locality or village. DNA was extracted from blood and tissue, and RAD sequencing was done. That is restriction site associated DNA extraction, DNA sequencing. That is a method of genome subsampling where SNPs, polymorphs, associated with restriction sites are sequenced. In this case, what I received initially when I joined was a VCF, which had about 349,000 SNPs. On getting that, the first thing that we did was to see how the different individuals are clustered related to each other within and between the localities. I did a PCA for that, and the blue circle on the left shows shows all the anciently invaded localities, and we see that it forms a smaller, tighter cluster. On the right are the recently invaded localities with much more differentiation comparatively. But overall, individuals generally cluster based on their sampling locality, except for 3 individuals here. The 2 in the center had comparatively lower quality data and were ultimately filtered out, so their positions could be attributed to that. While the one on the left here did not have low quality data, it was kept ultimately. And one potential reasoning is that it might be a recent immigrant from a different population or a different village, which was not sampled in this study. Then I used that, and this is where I started doing. I used that to do quality filtering of SNPs based on depth, the minimum percentage of individuals in which a locus has to be present so that it could be included, just getting the first SNP in every locus and that. And once the quality filtering was done, using the smaller filtered data set, I did a number of other analysis to understand clustering, structuring between the different localities to see how diversity differed between the localities, and finally, to see if there was isolation by distance. That is, if there was a correlation between geographic and genetic distances. Excuse me. After the initial data filtering, we were left with about 230 individuals. That's roughly 10 individuals less. The number of SNPs, though, was reduced by an order of magnitude. There it was about 350,000, but here it's 42,000. The maximum missing data that was allowed per individual, initially we tried to do a 20% filtering here, but in that case, one of the populations had reduced representation, very few individuals were left. And in order to be able to include all the populations here or all the localities here, we increased that, relaxed the filter to 50%. And finally, because this is more of a comparative study, we tried to keep only the locus loci, which were present in at least 80% of all individuals. Here we can see that in all the localities which were included, the mean number of samples per locus and the number of private alleles were approximately similar. Then I did a PCA on the filtered data set here. And one of the first things that was differences that we might notice is the level of clustering between this one and the unfiltered data set. It formed much tighter cluster in the previous case. However, there were also similarities in the general pattern, like the anciently invaded localities still formed a tighter cluster. But one of the recently invaded locality was formed a cluster away from the others. And this was the pattern which was there in the previous graph as well. A potential explanation could be from a previous study in this region, in the same region, northern Senegal. This was based on microsatellite data, and the localities marked with arrows are common in both the studies. Here we see that in St. Louis and in Dakar, there are two genetic groups of individuals. This was the first point of introduction, while this was the secondary introduction from here. And the expansion had started much later, so there's higher differentiation here. And we see that this locality, which is the one that clades out or separates out, has a considerable composition of both the genetic groups, while most of the others are majorly composed of just St. Louis. This is one potential reason why it might be separated out. Or a second one is that there were direct commercial links from Dakar to this region before the areas in its vicinity had road networks or links of that kind, which means that individuals in this region might have settled there much earlier compared to the other recently invaded localities. So this difference could either be because of the different invasion routes of these areas or because of their differential genetic makeup. We then did admixture analysis, where each of these lines represent a single individual and the proportion of their ancestry or genetic information from different genetic clusters. At k equals 6, that is with the assumption that all the individuals come from 6 different genetic clusters, the individuals separated out based on locality, that is sampling locality. At k equals 2, that is initially assuming that it came from just 2 genetic clusters, again, the recently invaded locality AEL separates out, showing that it's different from the others, and other lines of evidence, for example, their immune response has also showed that these individuals from this locality are different from the others. At k equals 3, we see that clustering mirrors the geographic distance. Sorry, that's at k equals 4. I'm sorry. At k equals 3, we see that the anciently invaded localities form a single genetic cluster, which is again in line with the PCA results, that it forms a tighter cluster. It's probably more closely related compared to the recently invaded localities. At k equals 4, there is a pattern of geographic distance. That is, the two localities in green here are closest to each other, and the two localities in blue here are closest to each other. We see a clear pattern of geographic distance. At k equals 5, finally, and which was also the group which was most supported and had the lowest cross-validation error, we see that 2 of the recently invaded localities still form a single genetic cluster, which means that there's potentially a role of geographic distance between the localities or between the individuals, which plays a role in their genetics. And to further confirm that, we did isolation by distance. Here, the x-axis shows log of geographic distance, that is between sampling points. The y-axis shows genetic differentiation, and the points represent all pairs of different individuals. Here, we wanted to see if this pattern, that is correlation of geographic and genetic distance, holds true when we compare the different localities or the different villages. And that's why we compared only the points to the right of the green line. Everything on the left has lower geographic distance, which potentially means that they are from the same locality and hence were ignored. We see that there's a positive slope, which means with increasing geographic distance, genetic distance also increases. We did mantle test, about 50,000 permutations, and it gave a significant value. What's interesting here is that between villages or between localities, dispersal of mice is majorly, if not entirely, mediated by humans. They get into vehicles, and that's how they are transported from one village to another. But human movement pattern is not as specific, or there's no specific pattern, and we did not expect to have a constraint on their dispersal because it was human mediated. But this graph shows that despite being dependent on humans, there are constraints on their dispersal between the different villages. We also did isolation by distance between the two regions, within the two regions. These are the localities in the anciently invaded areas, and these are the ones in recently invaded areas. And in all of these cases, the correlation is still positive and significant, which means the dispersal is still constrained here. Another reason why we did isolation by distance was to assess the dispersal, that is mean parent offspring distance, using the slope. And here we see that dispersal is higher in the recently invaded area compared to the anciently invaded ones. At the invasion front, which we assume the recently invaded areas to be, higher dispersal is advantageous to them because there are newer areas to invade, to colonize, and more movement would be advantageous to them in increasing their colonization abilities, which we see in this case compared to the anciently invaded ones. Sorry, and the last set of results here, this is within each of the different localities. We see that consistently FIS values show higher substructuring in the recently invaded localities. One potential reason could be founder events. That is, like I said, there was just one point of introduction and all the other localities or populations have come from previously existing ones. So with each successive event of founding a population, we would assume that substructuring would be higher, diversity would be lower, and that could be one explanation for higher structuring here. Or the second one could be that the size of these populations are still small and there is competition from native mice in this region, which is not the case in the anciently invaded ones. And that could also contribute to substructuring. One interesting thing here is that if there's higher substructuring, we would expect the heterozygosity levels to be lower, but it is either comparable or in some cases really high. There could be two potential explanations for this. First, that it is an effect of data filtering. And I think the two PCA plots are the biggest examples of how much effect filtering has had in our data set. It could either be that, or a more biological explanation would be that even though the population here is small, but it is probably founded by different existing populations. And hence there's substructuring, but overall diversity levels are still higher in this case. But this requires more work to figure out what exactly is going on here. So summing it all up, quality filtering has a significant impact, and any demographic inference which is drawn has to be done carefully based on the effect of the different filters which have been used. There is a very strong effect of geographic proximity that explains genetic clustering, genetic structuring within and between the localities. There's higher differentiation between localities in the recently invaded area. And this could either be because they have very small population size still, because it's very new, or that the reason is that their invasion history is comparatively much newer than the anciently invaded areas. Founder events might be responsible for reduced diversity because these are subsequent founder events. And finally, the advantageous trait of dispersion is higher at the recently invaded area, which probably means, which supports the idea that the expansion is still ongoing. A few ideas of future direction could be testing out the effect of other sets of filters, or maybe a larger range of parameters and order in which it's probably done to see how it affects the inferences. Change in effective population size over a temporal scale would give information on when probably these populations were established and how they are related to each other. Of course, a general one would be to increase more localities, especially in case of the recently invaded areas, because 2 of the 3 localities formed a single genetic cluster, which means they are probably a single population at this stage. And that leaves us with very little information to compare. So having more information in that region would give a better idea. And at this point, I have no idea how to do this, but one potential direction could also be modeling the process of invasion. And that's it. I thank all of these people, and I thank you for your attention. Thank you so much.